Hi everyone, it's Dr. Mickle again. I hope everyone is healthy and doing well. Today we're going to be looking at the continuation of the digestive system. So this is Digestive System Part 2, where we're going to be looking more at the functions of the different organs of your digestive system. We focused on the anatomy of the digestive system in the last video. Now we need to look a little closer at the functions of all of these organs. This figure nicely summarizes the major functions each organ is involved in, and we're going to work through all of these organs starting at the beginning of the alimentary canal and ending at the end of the alimentary canal. So this figure is nice because it's giving you all of the different regions of your alimentary canal plus the digestive accessory organs that are associated with that region. So you have your mouth and then the accessory organs like your salivary glands. You have your pharynx and your esophagus, which are parts of your alimentary canal. Stomach, part of the alimentary canal. Small intestine, and then your associated digestive accessory organs, such as your liver, gallbladder, and pancreas, and then your large intestine. To the right of each of these regions, you have the main functions that are occurring at that specific organ. And so if you look at your mouth, you have ingestion occurring. This is shown in light blue. Ingestion is only going to occur in the mouth. And then everything else is color-coded, and you can see that with the exception of two, ingestion and defecation, the activities are going to be kind of spread throughout the different organs. So in green on this figure, we have propulsion. And so propulsion is occurring in all of the regions, even if it's kind of hidden in the text for the stomach and your small intestine. Propulsion occurs in all regions of your alimentary canal. In dark blue, we can see you have mechanical breakdown of food, and this again is occurring in many of your regions. So it's occurring in the mouth, it's occurring in the stomach, it's occurring in the small intestine. The only places that it's not really occurring in would be your pharynx and esophagus, and then your large intestine. We do have chemical digestion going to occur in lots of different regions as well. You have it in the mouth, you have it in the stomach a little bit, the small intestine a lot, and then a little bit occurring in your large intestine. Absorption is only going to occur in specific areas. So absorption, there can be a few fat-soluble substances that can be absorbed in the stomach, but by far the site of absorption is going to be the small intestine, and then you have some in the large intestine as well. And then the last kind of category that we have, the only place that defecation is occurring is at the level of the large intestine. So this summarizes what we're going to look at in more detail throughout this video. And what I want you to do is, as we work through all of the organs and the accessory digestive organs, ask yourself which of the following 6 essential functions are occurring at each region. And so what we'll do is we're going to start at the beginning of the alimentary canal, the oral cavity or the mouth, walk through all of these organs and then the accessory organs associated with those regions. So your mouth or the oral cavity is going to be involved in ingesting food. And then we talked about your teeth in the last video. Teeth will help to allow for mechanical digestion, breaking down food into smaller substances so that there's a larger surface area for enzymes to work on. We talked about your accessory digestive organs that are your salivary glands that are all located outside of your oral cavity, but then you have ducts that would be carrying saliva into the oral cavity. Saliva is going to be very important. It is going to be mixed with your food as you chew to create a bolus of food or a blob of food that you then swallow, but saliva does more than this. So even though saliva is going to be primarily formed of water, it does also contain electrolytes and then enzymes like salivary amylase that can help to start the chemical breakdown of starches. It also contains another enzyme called lingual lipase, which will help to start to break down fats. Because of this, the presence of these enzymes in your saliva, chemical digestion begins in the oral cavity through these salivary enzymes. In addition, saliva plays a role in our sense of taste since it will help to dissolve food chemicals. But the oral cavity and the associated structures also play a role in propulsion. It plays a role in propulsion because you're going to have swallowing or deglutition occurring in the mouth. It's going to be initiated in the mouth, and swallowing is going to lead to food being propelled through the pharynx and the esophagus to reach the stomach. The start of swallowing is voluntary. You have conscious control over the start of swallowing, but once it's initiated, it is involuntary and automatic. You kind of can't have a half of a swallow, start to swallow and then stop. So there are two phases to the swallowing process, a buccal phase that occurs in the oral cavity, and then a pharyngoesophageal phase that is going to occur in the pharynx and the esophagus. So this figure and all of the images associated with this figure are showing you all phases of swallowing. The first image that you're looking at here is showing you just the events during the buccal phase. The figures that are associated with steps 2, 3, 4, and 5 are showing you portions of the pharyngoesophageal phase. So even though it's shown in 4 different images, this is all showing you 2 through 5 portions of your pharyngeal esophageal phase of swallowing. So if we look at your buccal phase, this is going to be the beginning of swallowing. Swallowing is initiated in the oral cavity when the tongue presses up against the hard palate. This forces the bolus of food, your blob of food, back into your oropharynx, and then it is going to be at this point that the pharyngeal esophageal phase begins. So for your buccal phase, this is going to be initiated by you. You have conscious control of it. It's going to start when the tongue presses the bolus of food against the hard palate and then forcing your bolus of food posteriorly into your oropharynx. At this point, this is going to be when your pharyngeal esophageal phase begins because the bolus of food is now located in your pharynx. When you have the pharyngeal esophageal phase beginning, this is going to lead to your soft palate and your uvula, so the soft portion of the roof of your oral cavity, and then your uvula. This will actually elevate to then block off the opening to the nasopharynx so that you don't have backwashing of food or drink into your nasopharynx and nasal cavity. When this occurs, you also have your larynx, which is going to be here in light blue. The larynx is going to rise, and the epiglottis is going to bend to close off the opening of your trachea, the glottis. At this point, the sphincter at the top of the esophagus, the upper esophageal sphincter, relaxes, and this will then open up the esophagus so the bolus of food can enter into the esophagus. The next phase of the pharyngoesophageal phase is going to be that the muscles of the pharynx are going to contract, and the contraction of the pharyngeal muscles will force the food into the inferiorly placed esophagus that is now open. After food enters into the esophagus, that upper esophageal sphincter is going to contract to prevent a backwash or regurgitation of food from your esophagus to your pharynx, hopefully. And then we move down to still these bottom images of 4 and 5 are still showing you portions of the pharyngeal esophageal phase. And so what you're looking at here is the movement of the bolus of food further down the esophagus, closer to the stomach. So at this point, if you look at step 4, you are going to have the adjacent sides of the esophagus contracting to cause that bolus of food to be moved down the esophagus towards the stomach. So this is considered peristalsis. Whenever you have adjacent sides of the alimentary canal contract, it's going to move your food forward, and it can be occurring in a very fast speed. The next portion is going to be what you look at in number 5. As that bolus of food approaches the stomach, you are going to have the cardiac sphincter, that sphincter around the opening of the stomach from the esophagus. That sphincter is going to relax, opening up the cardiac sphincter so that you can have the bolus of food entering into the cardial orifice, or the opening of the stomach. At this point, you are going to have the sphincter contract to then prevent regurgitation from your stomach into the esophagus. So once food enters into the stomach, that cardiac sphincter will contract to prevent any regurgitation. So now that the bolus of food is in the stomach, we can look at the activities that are occurring within the stomach. The stomach is going to first be involved in mechanical breakdown of food. So when the muscles of the stomach contract, it is going to slam that bolus of food up against the walls of the stomach, and the churning of your stomach and the slamming of the food up against the walls of the stomach and mixing with the gastric juices are going to lead to some mechanical breakdown of the food into a liquid, basically. You also, though, have chemical digestion occurring in the stomach. And so chemical digestion is going to be through enzymes or acids. And so this is involving the gastric juices that are produced by the stomach. So if we remember back to your video where we were talking about the anatomy of the stomach, we were talking about gastric glands, and that you have cells associated with your gastric glands that can produce hydrochloric acid, and then also the enzyme pepsinogen, which is activated to form pepsin. So these are going to be some of the enzymes and acids that we're going to worry about for chemical digestion of your stomach. Hydrochloric acid works by starting to denature or unwind proteins, and then pepsin is the activated enzyme that will help to start digest proteins. It's important to note, though, that chemical digestion, while it's occurring in the stomach, further chemical digestion must occur in the small intestine. So by the time food exits the stomach, it's not fully digested. You cannot absorb the components. You need further chemical digestion occurring, and this is going to occur in your small intestine. Lastly, the liquidized chyme that you have formed by the mechanical breakdown and mixing with the gastric juices and then some chemical digestion occurring in the stomach, that liquidized chyme needs to be propelled from the stomach to the small intestine. And this is something that has to occur in a very slow and controlled manner so that you are going to deliver the chyme to your small intestine and allow it to stay in the small intestine long enough for chemical digestion and absorption to occur there. So once food enters the stomach, it does need to mix with gastric juices. Gastric juices contain enzymes, acids, and hormones that are going to be important to the act of chemical digestion. The components of the gastric juices produced by the cells in the gastric glands found in the mucosal layer of your stomach. So this is a figure that we looked at in the last video, but it's showing you a gastric pit that would be one of these openings in the mucosal layer. And if you go down the gastric pit, it is going to lead down into a gastric gland. So here you have your gastric glands, and if we look at our gastric glands, we see that there's a change in the cells that are found in the gastric glands versus the gastric pits. Gastric glands have 4 different types of cells. There are mucous neck cells. We talked about these in the last video. Those are kind of the dark pink cells in this figure. And they are going to be involved in making an acidic mucus that we don't really understand its function for. Other cells that you find in the gastric gland will be your parietal cells. These we talked about in the last video as well. They're shown in blue. And then you have chief cells. The chief cells are shown in purple. Parietal cells are going to be responsible for forming hydrochloric acid and then also intrinsic factor. Chief cells, the ones that are shown in purple, are going to form the inactivated pepsinogen, which when it's present with hydrochloric acid will be activated to form the active digestive enzyme pepsin. But then chief cells will also produce lipases or enzymes that help with digestion of fats. In green, we have enteroendocrine cells. Enteroendocrine cells are going to be secreting a hormone called gastrin. And gastrin is going to be incredibly important for regulating when the stomach cells of your gastric glands secrete their secretions, and then when you will have contraction of the muscles of the stomach. So it's involved in regulating stomach secretion and motility. You produce quite a bit of gastric juices throughout the day. In one day, you make about 3 liters of gastric juices. And the secretion of these gastric juices is something that is very well controlled. You don't make it throughout the day. There's going to be careful regulation of when you produce these gastric juices. And this is because you don't want large amounts of acids and digestive enzymes in your stomach if there's no food in your stomach for it to work on. So the regulation of gastric secretion is going to have 3 phases. It includes a cephalic phase, a gastric phase, and an intestinal phase. The cephalic phase, when you see the word cephalic, think head. And so the cephalic phase is what is going to be what triggers gastric secretion, and it really is all in your head. So when you are looking at the gastric phase of gastric secretion, this is really a conditioned reflex that is initiated by thinking about food, smelling food, seeing food, or thinking about what you're going to eat for your next meal. So if you're baking cookies and you're waiting for them to come out of the oven, you're thinking about those cookies, you see them in the oven, and you're smelling them. All of these are going to trigger the cephalic phase of gastric secretion. And so all of a sudden you might feel your stomach kind of rumbling, you might start to salivate, and this is going to be because you have triggered this reflexive cephalic phase. So the thought, sight, and smell of food initiates a reflex where your vagus nerve is going to stimulate the cells in your gastric pits and gastric glands to start secreting their specific component of gastric juices. This is important because it ensures that there are gastric juices in your stomach ready to go before you've eaten any food, but you are planning on eating that food. So this is a way of making sure that as soon as food reaches the stomach, the gastric secretions are there and waiting for it. Once food enters the stomach, you then enter into the gastric phase of gastric secretion. As food fills the stomach will be stretched and then distended. And that is going to be a trigger to help to initiate your gastric phase of gastric secretion. Another trigger is going to be when you ingest food, there's a change in the acidity of your stomach. The presence of food will make your stomach less acidic. You also will have peptides that you've ingested present in the stomach that weren't there before. And so all of these are going to be kind of stimuli or triggers that will initiate your gastric phase of your gastric secretion. Gastrin has been released from the enteroendocrine cells, and the presence of this hormone will help to further stimulate the secretion of acids and enzymes. And this is all going to be going on during your gastric phase. So gastric phase, gastric, think stomach. It's going to be initiated when food arrives in the stomach. You have stretching of the stomach, distension of the stomach. You have a change in the acidity of the stomach. and all of these are going to be triggers for the gastric phase. This phase lasts about 3 or 4 hours and represents the time that food is going to remain in your stomach. This time is variable because it's based on the food that you have eaten. So if you have carbohydrate-heavy meals, that meal will spend less time in the stomach in comparison to a protein or fat-heavy meal. During the gastric phase, 2/3 of all gastric juices will be released. So this is going to be a pretty heavy phase for gastric secretion. Some of the stimuli that cause the gastric phase of gastric secretion deserve a little bit more attention. So we talked about enteroendocrine cells that are going to produce the hormone gastrin. Gastrin is going to be a hormone that when it is present and released, it leads to the release of more hydrochloric acid and enzymes from your parietal and your chief cells. But gastrin release is inhibited by low pH, which is pretty elegant if you think about it. If you have a low pH in your stomach, it means that you don't have food in your stomach. If you don't have food in your stomach, you really don't need gastric juices secreted because there's nothing to work on. And so the low pH is going to lead to gastrin being inhibited, and you would then have lower gastric secretions. But once you are eating a meal, the proteins you ingest will act as a buffer, and you will have a pH of the stomach that is going to be more basic than if the stomach was empty. This increase in the pH triggers the secretion of gastrin, which then causes more of your digestive enzymes and hydrochloric acid to be secreted. So gastrin, as well as other substances like acetylcholine and histamine, are going to all work together to stimulate parietal cells to release hydrochloric acid. And in order to have the maximum secretion of hydrochloric acid, you need to have all three, acetylcholine, histamine, and gastrin, present in the stomach to stimulate your parietal cells. The last phase of gastric secretion is the intestinal phase. This phase begins when chyme enters into the small intestine, hence the name. When this occurs, there's an additional release of gastrin into the intestine. This is a short-lived release of gastrin, but it's going to be as that chyme moves from the stomach to your small intestine, you get another burst of gastrin release. If you look at the delivery of the chyme from the stomach to the small intestine, this is something that is done in a very controlled manner. Chyme needs to be delivered in small amounts over periods of time, slowly, so that you have the chyme staying in the small intestine long enough to allow for chemical digestion and absorption to occur. An enterogastric reflex is going to help to ensure that the chyme stays in the duodenum long enough to allow for digestion and absorption. So when chyme is delivered to the duodenum, the small intestine is stretched. That stretch is the trigger that is going to initiate the enterogastric reflex, and at this point it's going to inhibit gastric secretion, inhibits gastric secretion, and then also the motility of the stomach. At this point, we have chyme in the small intestine. It is going to be partially digested carbohydrates and undigested fats. So a lot of chemical digestion still needs to occur, and the sheer majority of this chemical digestion is going to occur in the small intestine. So because you need quite a bit of digestion to still occur, and then you need absorption to occur, the chyme is going to stay in the small intestine for about 3 to 6 hours. During this time, chemical digestion is occurring, and you have absorption of most of the nutrients and a lot of the water through the villi, and then having it dumped back into the bloodstream. The small intestine is the major site of chemical digestion and absorption, and it also plays a role in propulsion. Associated with your small intestine will be a few of your accessory digestive organs. And if you remember back to the last video lecture, we talked about how the duodenum has bile and pancreatic juices delivered directly to it. Bile is produced by one of those accessory digestive organs, the liver, and then it is stored temporarily until it's needed in the gallbladder. You have a series of ducts that will drain your liver and the gallbladder called the biliary tree that's responsible for delivering this bile to your duodenum. And your biliary tree are going to be all of these green ducts that you see associated with your gallbladder and your liver. The function of bile is to emulsify lipids. And basically, you want to think of bile as being a pre-treater that helps to allow for digestion of lipids to occur more easily. So you have your bile pre-treating the lipids so that they can be broken down and digested by enzymes later. Lipids that enter the small intestine are basically not digested at all, and they're not small enough to be absorbed. So digestion of lipids occurs in the small intestine and starts with those bile salts. Bile salts are going to help to break down the big droplets of fat into smaller droplets so that you're increasing the surface area for enzymes to actually digest your fats. So bile, it's produced by your liver. It is going to be stored in your gallbladder, delivered to your duodenum via your biliary tree, and the function of it is to emulsify fats, break it into smaller droplets so that there's more surface area for your bile to work upon. It is not chemically digesting your fat. Pancreatic juices are also delivered to the duodenum, and in this case, your pancreatic juices are going to be drained from your pancreas through a long tube called your main pancreatic duct. The main pancreatic duct is then going to open and empty into your duodenum. Pancreatic juices contain enzymes such as pancreatic amylases that will digest starches, pancreatic proteases that will digest proteins, pancreatic lipases that digest fats, and then nucleases that break down nucleic acids. Pancreatic juices, along with having enzymes, will have buffers that will help to neutralize the acidic chyme that's been delivered from the stomach to the small intestine. In order for there to be maximum digestion and absorption occurring in the small intestines, three things must happen. First, you need to have an acidic hypertonic chyme delivered slowly from the stomach to the small intestine. You also need bile, pancreatic enzymes, and then the buffering bicarbonate ions delivered from the accessory digestive organs. Last, you need to mix the chyme and these digestive juices. The mixing is going to be helped out with those circular folds that slow the process of chyme moving through the stomach. And then you also have associated with those circular folds all of your villi and microvilli. On top of having your contents kind of spiraling through the circular folds as it moves through the small intestine, you are also going to have mixing of the contents occurring via segmentation. Segmentation is going to be the most common motion that occurs in the small intestine, and it involves mixing or moving contents forward and back, and slowly propelling it towards the ileocecal valve. Segmentation occurs when non-adjacent segments of the small intestine alternatively contract and relax. This is what makes it different than peristalsis. In peristalsis, it's going to be adjacent segments of the small intestine alternatively contracting and relaxing. So segmentation is going to occur when those non-adjacent segments of the small intestine contract or relax. There is a link to a little video that shows you segmentation occurring in the small intestine that might be able to help you visualize how you would have mixing of the contents as you have segmentation occurring. Take-home message about segmentation, though, is it's the most common motion of your small intestine. It is slow. It will allow for mixing of the contents, and it would be slowly progressing the contents towards the end of the small intestine. Peristalsis is going to be another activity that occurs in the small intestine. It differs from segmentation in that you have adjacent segments of the small intestine alternatively contracting and relaxing to push contents down the small intestine in a much faster manner than your segmentation. Peristalsis is stimulated by the hormone motilin, and this is going to be secreted at the end of the intestinal phase. And peristalsis occurs in waves about every hour to an hour and a half. The waves of peristalsis migrate, so you would have a wave that would start and then it would stop, and the next wave would begin distal to the wave that preceded it. In this way, remnants in the small intestine are going to be moved through the small intestine towards the large intestine. In order for contents to move from the duodenum to the ileum, it is going to take about 2 hours. Once you have contents making it to the distal end of the ileum, the end of the small intestine, you are going to have the ileocecal sphincter at the junction of the ileum and the cecum relaxing. When this valve relaxes, it is going to allow your contents to move from the small intestine into the first portion of your large intestine, the cecum. It occurs when a gastroileal reflex makes the force of segmentation in the ileum greater, and it also is going to be triggered by gastrin that increases the motility of the ileum. After the contents of your small intestine are emptied into the cecum, the contents are going to exert a back pressure onto the ileocecal valve and shut the valve to prevent kind of a backwash of now large intestine contents into your small intestine. Once contents make it into the large intestine, it is going to stay there for about 12 to 24 hours. There is no digestion of foodstuffs occurring via enzymes produced by you in the large intestine. The only breakdown of food that is occurring in the large intestine is through enteric bacteria, or your good gut bacteria. Your good gut bacteria will produce vitamins that are going to be absorbed into the bloodstream through the small intestine, and so that is important. This is why if you have a lot of antibiotics and you kind of destroy your good gut flora, you could have GI issues and you could even have issues with absorbing vitamins. The other types of absorption that are occurring in the large intestine will be some electrolytes, specifically sodium and calcium, will be absorbed from the large intestine. And then really the last component, probably the one that's most important for the large intestine, will be you will have water absorbed. As water is absorbed, the feces are compacted. Another major function that occurs in the large intestine would be propulsion of the feces through the tract towards the rectum and the anus, where then defecation occurs. The colon does not play a huge role in the digestion of food and absorption of nutrients. A lot of that, the sheer majority, will be occurring in your small intestine. And so your large intestine isn't actually considered essential for life. You can have large amounts of your colon removed without life-threatening effects. For motility of the large intestine, there are going to be two types of contractions that occur in your large intestine. There are going to be haustral contractions that are caused by haustra sequentially contracting in response to that single haustrum being full and distended. These haustral contractions are going to cause kind of slow segmentation movements. The other contraction that occurs in your large intestine is going to be the result of a gastrocolic reflex. So the name is telling you gastro, stomach, colic, colon. So the gastrocolic reflex is a reflex that's triggered by the presence of food in your stomach. A gastrocolic reflex triggers 3 to 4 slow but powerful peristaltic waves in the colon per day, and these waves are going to move contents longer and quicker than the movement caused by the haustral contractions. These movements are referred to as mass movements, and they are responsible for moving the contents towards the end of the large intestine, towards your rectum. So as we went through our tour of the different regions of the alimentary canal, we looked at areas that were involved in chemical digestion. Chemical digestion is the breakdown of macromolecules into monomers that are small enough to be absorbed. It occurs via enzymes that are produced by the alimentary canal organ itself or accessory digestive glands associated with that organ. We saw that the mouth and the stomach play a small role in chemical digestion. The small intestine is going to play a major role, and the large intestine plays a minor role with the involvement of bacteria. What we have remaining to look at is how different macromolecules require different enzymes to be broken down, and how different foodstuffs are going to be digested differently, and how those digested foodstuffs are absorbed. So this figure nicely summarizes how different foodstuffs are digested and what enzymes are involved in digesting that specific type of food. It also shows you where those enzymes are working, whether it be in your mouth, the stomach, small intestine, or large intestine. And then lastly, it shows how absorption of the end products of digestion occurs. This is a big figure, and there's a lot going on here, but what you should be familiar with is what enzyme is involved in the digestion of each type of food, as well as where those enzymes are acting, and then how absorption is taking place. So you need to be familiar with those concepts for carbohydrates, proteins, and fats. Let's start with the simplest foodstuff and work our way towards the more structurally complex food types. So carbohydrates need to be broken down to monosaccharides or monomers that can be absorbed. Breakdown of carbohydrates or starches starts in the mouth through salivary amylases. These enzymes are going to break starches down into smaller fragments, and this continues in the stomach until the stomach acids break down your enzymes. In the small intestine, pancreatic amylases and brush border enzymes further break down the digestion of starches into monomers that can then finally be taken up by the cells of the epithelium of your small intestine. They are going to be co-transported with sodium, and this is going to be how they are going to enter into the cells. It is then going to be passively transported into capillaries and into your circulation. So moving on to proteins, we're looking now at the middle column of this figure. Proteins need to be broken down to their amino acid building blocks in order to be absorbed. Digestion of proteins begin in the stomach with pepsin and hydrochloric acid. But these are not going to fully digest your proteins into small enough sizes that they can be absorbed. Further digestion needs to occur, and this is going to occur in your small intestine. When you get to your small intestine, you are going to have pancreatic proteases and brush border enzymes that are going to work on the smaller subunits of proteins so that you break them down to amino acids. Amino acids are then going to be taken up by the epithelial cells of the small intestine via co-transport with sodium and then passively transported into the capillaries. So the absorption is going to be similar to what you saw for your carbohydrates. Last but not least, we have lipids. Lipids are structurally complex, and they're also insoluble in water. You do have some chemical digestion occurring in the oral cavity via lingual lipases, but these lingual lipase enzymes can only work on the exposed surface of lipids. The action of the lingual lipases are not going to be enough to break down lipids, typically ingested as triglycerides, into small enough components that can be absorbed. You're going to need your bile and pancreatic lipases in order to break down your lipids into small enough components to be absorbed. So bile and pancreatic lipases are going to be working in your small intestine. Bile salts are going to emulsify or pre-treat lipids before they can be broken down by your pancreatic lipases. And how bile salts work is they break up the big, large fat globules into smaller droplets. This is not digestion. There are no bonds being broken by bile. Instead, bile works by breaking the large fat globule into a smaller globule to increase the surface area on those smaller globules so that pancreatic lipases can do their work. Pancreatic lipases break down lipids into fatty acids and monoglycerides, which can then interact with bile to form these little complexes of lipid and bile called micelles. The micelle can then come into contact with the epithelium of the intestine, and then the lipids diffuse across the cell membrane to enter into the intestinal cell. Once you have your lipid components in your intestinal cells, they are going to be converted back into triglycerides coated with proteins to form a complex that is going to be released by the cell via exocytosis. At this point, you now have the triglycerides in the interstitial fluid, where you will have them transported to the general circulation via the lymphatic system. Lastly, though absorption occurs in the small and the large intestine, it is by far more important in the small intestine. Almost all of the nutrients of the food you eat, about 80% of the electrolytes, are going to be absorbed in the small intestine. Most of this absorption is occurring before contents reach the ileum. So think you're really dealing with your jejunum. The small intestine is responsible for absorbing 95% of the water from intestinal contents. And if you think of 9 liters entering the small intestine a day, that's a lot of water absorbed via osmosis in your small intestine. The majority of that remaining 5% of the water is going to be absorbed by the large intestine. So if you look back at what the function of the small intestine is for absorption of nutrients and water, this explains why celiac sufferers have problems with nutrition and also GI issues, because in celiac disease, you will have the villi in your small intestine either laying down and not being able to function or completely destroyed, and then they would not be able to allow for absorption. So you actually have malnourishment in people who have untreated or unrecognized celiac disease, because even though they're ingesting food, they are not able to absorb those nutrients because the villi are not functioning in their small intestine.